Hello friends, welcome to BMH Learning. So we are discussing about lipid biosynthesis. And this video specifically deals with synthesis of triglycerides, also called triacylglycerols. To make things easier, the synthesis of triglycerides can be divided into steps. The first step is the synthesis of glycerol 3-phosphate. So this is the structure of glycerol 3-phosphate. The glycerol 3-phosphate can be formed in two ways. You must remember about glycolysis. So when we break glucose, we get this molecule as an intermediate of glycolytic pathway. This is named as dihydroxyacetone. Dihydroxyacetone is the first molecule from which glycerol 3-phosphate is made. And this task is carried out by glycerol 3-phosphate dehydrogenase. Because this is a dehydrogenation reaction, we need a reducing agent in the form of NADH. The second source of getting glycerol 3-phosphate is glycerol itself. So we can directly attach a phosphate group at the third position of glycerol to make it glycerol 3-phosphate. So for that, we have to use an ATP molecule. Because we are transferring a phosphate group, we need a kinase which is named as glycerol kinase. Once the glycerol 3-phosphate is formed, we move to the next step, which is the attachment of fatty acids to glycerol 3-phosphate. In this molecule of glycerol 3-phosphate, there are two positions where fatty acids can be attached. Let's do at carbon number 1 first. So, we add R1-acyl-CoA, which is nothing but an activated fatty acid. The reaction results in the formation of glycerol 3-phosphate, now with a fatty acid at carbon number 1. The name of this molecule is lysophosphatidate, and this is done by acyltransferase. Now, once this is done, we will repeat the same procedure for carbon number 2. So we will take another acyl-CoA in the form of R2, which is different than R1 in terms of having more or less carbon atoms. So R2 activated fatty acid is attached at carbon number 2. The molecule we are getting is called phosphatidate. The reaction is catalyzed by acyltransferase. This compound is also famous with the name diacylglycerol 3-phosphate. Now this is a point from where two different types of lipids are produced. The one which we are talking about is the triglycerides, and the other one which are present in membranes and are called as phosphoglycerides. The synthesis of phosphoglycerides will be done in next video. For now, just concentrate on triglycerides. So the third step is the formation of triglycerides. So we have formed diacylglycerol having a phosphate group at third position. Now look at this molecule. It's similar to the phosphatidate, except it has one more fatty acid at third position. This molecule is the one which we want to make, because this is a triglyceride. So what we are going to do, let's remove the phosphate at third position. To get rid of phosphate is a work which is accomplished by phosphatase. So we bring phosphatidate phosphatase. This enzyme activity releases phosphate and we get diacylglycerol. Finally, we treat the carbon number 3 in a similar fashion as we did with position number 1 and 2. So another molecule of fatty acyl-CoA in the form of R3. And this will lead us to the product which looks exactly like triglyceride. The reaction is carried out by acyltransferase. This was all. Thanks for watching.